This video is about aerobic cellular respiration. Aerobic cellular respiration is a series of metabolic reactions to create energy. It takes place in the mitochondria of the cell. We will be creating energy from the molecule glucose in 4 steps. The first step is glycolysis. The second is pyruvate oxidation. Then it's followed by the citric acid cycle, also known as the Krebs cycle. And finally, it'll finish off with the electron transport chain. We will begin with glycolysis. There are 2 phases in glycolysis. The first is called energy investment. The reaction begins with the molecule seen here, a 6-carbon sugar named glucose. This comes from the food that you eat. The glucose will then react with a molecule known as ATP, or adenosine triphosphate. It is called that because there are 3 inorganic phosphate molecules attached together. The reaction happens when an enzyme comes and breaks apart the bond between 2 of those inorganic phosphates. That phosphate group will then go on to attach to the glucose molecule, removing the hydrogen, and our ATP molecule will now become ADP, or adenosine diphosphate. We have just created a new molecule known as glucose 6-phosphate. This reaction is called a phosphorylation reaction. In the next step, the glucose 6-phosphate will now be rearranged into its isomer, fructose 6-phosphate. This is called an isomerization reaction. In the third step, another phosphate group is broken off the ATP molecule and will attach to fructose 6-phosphate. This creates another ADP molecule, and we now have fructose 1,6-biphosphate. This is another phosphorylation reaction. The fructose 1,6-biphosphate molecule will now be split into 2 different molecules. The first one being G3P, or glyceraldehyde 3-phosphate, and the second is DHAP, or dihydroxyacetone phosphate. They are both 3-carbon molecules and are very similar in their structure. This is known as a lysis reaction. Before we move on to the second phase in glycolysis, we are going to rearrange our DHAP molecule and turn it into another G3P. At the end of energy investment, we have now lost 2 ATP. And with our 2 G3P molecules, we are now ready to move on to the next stage, energy payoff. Because of the 2 G3P molecules we have created in the last phase, from now on, there will be twice as many products, even though you'll only see one on the screen. To begin the energy payoff phase, 2 electrons and 2 protons will be removed from the G3P. Some of the energy released in this reaction is trapped by the addition of an inorganic phosphate group that comes from the cytosol, not from another ATP. It will then attach onto the G3P molecule. An NAD+ molecule will then come along and accept 2 of the electrons and 1 of the protons. This will create the molecule NADH, while the other hydrogen will be lost to the cytosol. The G3P will now turn into 1,3-biphosphoglycerate. This is known as a redox reaction. In the 7th step, one of the 2 phosphate groups on the 1,3-biphosphoglycerate will be transferred to ADP, and it'll produce the high-energy molecule ATP. This is called a substrate-level phosphorylation reaction. And our 1,3-biphosphoglycerate molecule will now be turned into a 3-phosphoglycerate molecule. In the next step, we'll see a mutase reaction. 3-phosphoglycerate will be rearranged into 2-phosphoglycerate because of a shift of the phosphate group from the 3-carbon to the 2-carbon. In the 9th step, electrons will be removed from one part of the 2-phosphoglycerate and delivered to another part of the molecule. There is also a loss of water. This is called a redox reaction. The 2-phosphoglycerate will now be changed into a phosphoenol pyruvate. In the last step of glycolysis, the remaining phosphate group will be removed from the phosphoenol pyruvate and be added on to ADP to form another ATP molecule. Here we have the final product of glycolysis, known as pyruvate. Along the way, in the energy investment phase, we lost 2 ATP molecules. And in the energy payoff phase, we gained 2 NADH molecules and 4 ATP molecules. This gives us a net total of 2 ATP and 2 NADH molecules at the end of glycolysis. We now move on to the next stage of cellular respiration, known as pyruvate oxidation. The purpose of this stage is to get the pyruvate into the mitochondrial matrix. In order to do that, the pyruvates that are produced in the glycolysis must pass through both the outer and inner mitochondrial membranes. Pyruvate can diffuse easily through the outer membrane because of the large pores. But it'll need to undergo 3 reactions in order to enter the matrix. The first is a decarboxylation reaction. The carboxyl group of the pyruvate will be removed to form CO2 as a waste product. This is actually a third of the carbon dioxide that we exhale. The second step is a dehydrogenation reaction. 2 electrons and a proton will be transferred to NAD+ to form another NADH. Another H+ molecule will be lost into the solution. We now have an acetyl group left. Lastly, the acetyl group reacts with a sulfur atom of a coenzyme A, forming the high-energy intermediate acetyl-CoA. It is now ready to be transported across the inner mitochondrial membrane. We now move on to the third stage called the citric acid cycle, also known as the Krebs cycle. It consists of 8 enzyme-catalyzed reactions, 7 of which take place in the matrix and 1 binds to the matrix side of the inner mitochondrial membrane. It is called a cycle because the final products of the last reaction will be reused to start the cycle over again. The first step takes our 2-carbon acetyl group carried by coenzyme A and transfers it to oxaloacetate, forming the molecule citrate. The coenzyme A is lost and water, or H2O, is added. In the second step, citrate will be rearranged into its isomer called isocitrate. In the third step, another carbon group will be removed and released as the waste product CO2. The isocitrate will then be oxidized to form NADH from NAD+, and another proton will be lost. We are now left with the 5-carbon molecule alpha-ketoglutarate. And the same reaction will occur again. Alpha-ketoglutarate will be oxidized, NAD+ will be reduced to NADH plus a proton, and will lose another carbon as CO2. The only difference is this time, coenzyme A will be added into the cycle to form the new molecule succinyl-CoA. In the 5th step, the CoA will be released and will also release energy. This energy converts GDP into GTP, which in turn converts ADP into ATP by substrate-level phosphorylation. This is the only ATP made directly in the citric acid cycle. The succinyl-CoA now turned into succinate, a 4-carbon molecule. The succinate will now be oxidized again. 2 electrons and 2 protons removed from succinate will be transferred to FAD and will produce FADH2. We'll now be left with fumarate. Through the addition of a water molecule, fumarate now turns into malate. And in the last step, malate will be oxidized and reduce NAD+ to NADH and H+. We now return back to where we started with the oxaloacetate, and we're ready to finish the last stage of cellular respiration. The electron transport chain. This stage extracts potential energy from NADH and FADH2 and makes it available for the synthesis of ATP. If we take a closer look, we can see where this is all happening. We can now see the cytosol of the cell, the outer mitochondrial membrane, the inner membrane space, the inner mitochondrial membrane, and finally the mitochondrial matrix. The electron transport chain takes place here, in the inner mitochondrial membrane. It consists of 4 protein complexes. The first is called NADH dehydrogenase, it is made up of multiple proteins and accepts electrons from NADH. The second is a succinate dehydrogenase. It is a single protein and accepts electrons from FADH2. The third complex is called the cytochrome complex, and the fourth is called the cytochrome oxidase. The last structure there is the ATP synthase, which we'll talk about later. Electrons will be passed between these complexes by shuttles. The first is called ubiquinone. It is a hydrophobic molecule found in the core of the membrane, and it receives electrons from both the first and the second complex. As you can see here, the NADH molecule gives up its electron to the first complex and is oxidized to NAD+. The electron is then passed along to the ubiquinone shuttle. And as that happens, the energy given up from the electron pumps the H+ ion through the complex to the intermembrane space. The UQ shuttle now arrives at the second complex and picks up another electron from FADH2. The shuttle now holds 2 electrons as it moves on to the third complex. And as before, another H+ ion is pumped into the intermembrane space. From there, the 2 electrons will now be carried into the fourth complex through another shuttle. This one is called cytochrome C. It carries the electrons through the intermembrane space to the fourth complex. In the fourth complex, the 2 electrons now provide energy for the formation of H2O. This happens because the increase in electronegativity among the complexes. Each complex is more electronegative than the last, until it reaches the final electron acceptor, which is the most electronegative of them all, oxygen, or O2. This system happens almost instantaneously because of electronegativity. Since oxygen is more electronegative than complex 4, it will pull away the electrons. Now complex 4 is oxidized, but because it's more electronegative than complex 3, it will attract the electrons from the third complex. The third complex then takes the electrons from the first complex, and the first complex takes the electrons from NADH. In the end, for every O2 gas molecule we breathe in, 4 electrons are pulled through the electron transport chain, and 2 water molecules will be produced. You must be wondering what the ATP synthase is there for. It has to do with the real way that the ATP is produced, through chemiosmosis. If you recall from before, every time an electron was taken off a carrier molecule, a proton was carried through from the matrix into the intermembrane space. This has created an area of high concentration in the intermembrane space and low concentration in the matrix. This is called a proton gradient, and it is a form of potential energy. Because the protons are charged, they repel each other and are attracted to the more negative interior of the matrix. This combination of events has created a proton motive force. And the ability of cells to use the proton motive force to do work is called chemiosmosis. We're going to use this energy to convert ADP into ATP. The basal unit, embedded in the inner mitochondrial membrane, is going to act as a channel for the protons to freely pass through. The proton motive force moves the protons in the intermembrane space through the channel, down their concentration gradient, and into the matrix. This flow of protons powers ATP synthesis by the headpiece, which is sticking out into the matrix. When 3 protons are added, the headpiece will start to spin, kind of like a motor. This energy is then used to synthesize ADP into ATP. Theoretically, we should be able to yield 34 ATP. In the end, glycolysis produced 2 ATP molecules. the citric acid cycle produced 2 ATP molecules from the 2 pyruvates that went through it, and the electron transport chain and chemiosmosis produced 34 ATP. This makes a grand total of 38 ATP.